Well, I mean, thank you very much for this nice introduction, Carlos and Suzanne. And what I am going to do today is that as the institution that are in charge of work package 3, I'm going to tell you our effort to uh, modify the surface of these nanomaterials, which are practically used in industry, and to see how the surface chemistry affects their toxicity, or the goal is to reduce their adverse effects through surface chemistry, of course. And then do the testing, in vitro screening, and then see how the in vitro screening is a valuable tool to understand their toxicity. Here's the outline of my talk. I don't know why this is going faster, but first I'm going to show you nanomaterials and their properties, and I'll give you some brief information about their safety. Then I will mention briefly about the safe by design concept. Then the nanomaterial toxicity and the surface chemistry relation. Then of course, I need to show you some case studies, some materials, and how surface chemistry is affecting their behavior in biological system and environment. Then these are basically, we are doing two types of in vitro tests. The first group is, to check with the eukaryotic cells, related to human health. The other is the environmental concern, and that is, it's called ecotoxicity issues, and I will conclude my talk. Well, I think, the audience and everyone knows who are in this field that there is a vast majority of the vast number of nanomaterials available to us. Any material, technically can be brought into nano size to explore their function. Okay, so I'm not going to go through this, but if you are in this field, or if you are even related to this field, and I'm sure you know the diversity there. There's a huge number of particles out there. Okay, for example, this slide is just showing some TEM, SEM images from our studies, and then as you see, they are even available for a variety of different applications because of their functions, because they show unique properties in new, maybe novel products that might be useful for us. And this is the list of nanomaterials that we studied from the companies who are part of this project. Okay, as you see, I think Carlos nicely mentioned all these companies and their materials, and the nanomaterials we used from them, from their production. And as you see, it's just, again, it's a huge diversity here, different forms, different applications, and, and the different sizes and so forth. Okay, so, because I'm just trying to tell you that there is a huge variation in the nanomaterials. Okay, so what is the concern here? Concern is because if these materials are used in any application, at one point, of course, from the humans will be affected. Also, next, environment will be affected, because we first, as humans, we use a product, then where these materials go, they go to environment, right? So there are a lot of different scenarios, they can affect health and environment. So, for example, in biological systems, once they make into the human body, they may go through many different barriers, and they may damage the organs. And once they make into the cells, they can actually affect the cellular events that, resulting in different, situations, like including even cancer. So we need to understand how these materials behave in these biological systems. Okay, so here's the issue is that because can we make these materials safer? Okay, because we are using them, right? Okay, then we know that they are useful. Can we make them safer? So that is the whole idea. this safe by design idea is not new, but its application in nanotechnology is new. So actually, this is, you know, the definition of safe by design is to start, you know, all the safety concerns from the scratch during the R&D stage. Then, you know, go through all the steps, you know, from the R&D stage to the release into environment. So, for example, if you build a building, so you need to take care of all the concerns, because the safety concerns, because people will be moving, going into the building, living in the building, right? We don't want to collapse them in 2 days later. So you need to take all the precautions before you start making the building. So the same, you know, same idea here, because once we start, you know, considering all the function, okay, why we are aiming to use, you know, in the application, we need to start from the scratch, from the design stage, to make this material safer, and even after their use in the application. So, well, I mean, probably, I mean, I'm assuming that you know some about the, because if you are listening to this webinar, you know nanomaterials. Nanomaterials, from chemistry perspective, it is not that easy. You know, if you're designing something, it's, for example, carbon nanomaterials. You make them, right? I mean, there's not much control until you make them. So the size, maybe you have a control. I mean, the shape, maybe you, I mean, these are difficult, you know, things, the shape and size, for example, to control. So because you make them, because they are nice materials and they have nice functions, for example, electrical or some, you know, carrier functions or such. So because you cannot play with their size and shape too much, because they're a carbon-based material, you just make them. So at the end, I mean, this is just one example, but if you consider other nanometers, you don't have much control, right? Because even you change the size, you change the function. So it is not much room there to play with their size and shape to make it safer. So what happens is that, here's the deal, because the idea is to change the surface chemistry, because this might help the interaction of these materials with the environment wherever they are residing in the matrix, which is, you know, in this case, it's biological and biological medium, biological environment here, right? Because this is the question to why is because surface is the interface to the outside, mainly this, you know, the biological medium. Okay, so, well, I mean, once you try to change the surface chemistry, you need to take, you know, you consider a lot of parameters together. So which surface chemistry you're going to use, which molecules you will be using, what polymers you will be using. So then are they biocompatible, okay? Are they, once you make the surface modification, are they stable in the formulation? And then is it, you know, chemically suitable for that kind of application by considering your application? How about the cost? It's really important, right? Because if you're going to make a mass production, is it really possible, you know, to use, you know, possible to obtain it in a low cost? Because it's going to cost you, if a product is going to cost more, you don't want to use it, because, you know, no one is going to buy it, right? Also, you need to consider the toxicity concerns. For example, what is going to happen to this surface chemistry? Is it going to be stable on the surface? Is it going to be leaving, or if once it leaves, it is going to be toxic or not? And there are a lot of issues. If, for example, aggregation is another problem. Okay, after taking care of all these, you know, concerns, now you need to consider the surface modification. So what happens is that surface modification is another issue, because you are going to again use chemicals, right? So you need to pick the best strategy to not to contaminate your nanomaterials, because, you know, all chemicals that are used in this, you know, modification strategy can affect the toxicity. Okay, is it going to, are you going to use just a simple physical coating, or just attach the molecules that you selected through certain chemistry? Is this chemistry available? And all these, you know, the question should be answered to develop a strategy, modification strategy. So now I'm switching to the point that we are, how we handle this. From the experience, our experience, and also from the literature studies, and what we did is, I came to the conclusion that small molecules, biological molecules, okay, with non-toxic biological molecules such as glycine and glucose and these monosaccharides and amino acids, and they're good candidates because they will not alter the function of nanomaterials as well. Because just changing the surface without changing too much of the other materials, I mean properties of materials. Okay, so then next step was to modify them. Today I'm going to show you especially these carbon-based nanomaterials later on with the in vitro toxicity testing. But here this is showing how carbon-based nanomaterials are modified, the chemistry and their modification strategies. But I included ethylene diamine to see if I change the surface, because if your glycine, makes the surface negatively charged, ethyl diamine makes the surface positively charged, and the glucose is just, of course, it's neutral. To see these effects that I made the changes later on, I'm going to focus on this. But before I go on, Carlos nicely presented all the companies who provided diverse nanomaterials with diverse surface chemistries. So for diverse applications, okay? It is not that easy because I'm trying to make a point here later on when I start talking about the in vitro toxicity. I will, come back to this point because the diversity is important. Of course, as I said, the function of the nanomaterials are really important as it comes to the modification, everything, all these things that I talked about, is really, the important not to change the function of the nanomaterial, because in the application at the end of the day, you need to use this in the product, and you want that your product is going to be functioning the same. So this is an example. For example, this is sulfur-doped titanium silicon oxide nanoparticles used by Creative Nano in Greece. They use these materials for photocatalytic paint, for example. Okay, so now I'm going to go into this nanotoxicity the tests. Okay, there are two in vitro tests. These are done in the laboratory. Okay, so the first is concerning the human health. The other is the environment. This is I'm going to just show you which tests we use in this project to concerning the human health. These are in viability tests, VST8 assay cell viability test we used, and the other test we used was the COMET assay for DNA damage. And the for environmental tests, it's just a little bit complicated, and we added more types of different tests that I'm going to, so ZSA and alga growth inhibition test and earthworm acute toxicity test, and bioaccumulation tests. Then I'm going to talk about this, possible issues there as I move on, and to tell you the complete story here. Okay, here's the question. Maybe you may be thinking, okay, we are doing this in vitro tests. Okay, then we are concerned about the environment. Of course, a different story, but the human health is in the real, biological system in the human body. Are they going to be the same, their behaviors and their effects? There's an effort to correlate the in vitro and in vivo tests. Okay, so I mean, it is. Sometimes there's a good correlation, sometimes not. Because why this is so? Because nanomaterials' behavior in the biological systems is truly, I mean, how they behave in a matrix truly depends on the matrix. Okay, if you change the environment, for example, if you have a PBS buffer, if you put these materials into cell culture medium, it behaves differently. If you throw them on the soil, it's just a different story. So that's why, if, for example, cellular study is just, it's just more maybe compatible. I mean, there's a correlation with this, in vitro and in vivo. Okay, there's some, literatures that are arguing that, it is possible or not possible, but there's some good correlation that I would say. The other point is because I told you about the diversity of these nanomaterials, because you cannot test them in vivo, all these diverse nanomaterials out there. So all the way to test them in the laboratory through high throughput testing. Okay, so then of course the cost and time, because you cannot just go and try on animals, because it's gonna cost you more and it's gonna cost you more time. So there's an effort to move from in vitro, to develop in vitro tests which mimics the in vivo systems, such as 3D cell cultures, and then also some microfluidic system and so forth. So that's why it is, in this project we focus on in vitro. We don't want to, because we don't have the capacity to test that diverse nanomaterials in vivo testing. So now I'm going to show you some results with the in vitro toxicity testing. Just we selected 3 cell cultures. These cell cultures, first of them HSF, is human skin fibroblast tissues, cells, and the other is human bronchial epithelial cells, and the other is the macrophage. Why we selected these cell lines? Because considering the exposure, nanomaterial exposure, how people are exposed to these nanomaterials. For example, if someone is making these nanomaterials, they probably exposed to these materials through the skin, or they can aerate, they can breathe these materials. Then of course, once these nanomaterials make into the human body, what happens is that they cause an inflammation and immune response. That's why we selected these 3 types of cell types. Then here's the results. For example, I show you this sulfur doped titanium oxide, silicon oxide nanomaterials. As you see here, this material is just typical. Very typical. And with the in vitro testing, they show some toxicity, some don't show, depending on the sulfur content in these nanomaterials, because that is the concern. Because we usually know that titanium and silicon nanoparticles, they are not very toxic. They're okay, and even they are studied for drug delivery applications. But if sulfur is doped into it, so what happens? Do they cause problems for example, when they interact with cells? It is quite, showing that it is not really toxic to these cells with the time and concentration dependent study. This another the nanomaterials from Laurentio Technologies. We tested the silicon oxide nanoparticles. On the surface, in one type of particle, there's only hydroxyl groups. In the second type is amino groups. This, the bottom of the slide is showing their characterization. Of course, we had to characterize fully to make sure that this material is really what it is meant. So as you see here in the different cell lines, it's obviously silicon oxide nanoparticles in this case is showing toxicity on these cell lines we tested. Because as I said earlier, why silicon oxide nanoparticles are not toxic, but in this case, it is toxic because I told you there's a huge variation from synthesis. Even synthesis makes a big difference because who knows what kind of chemicals they used is causing this toxicity. Is it just the nanomaterial itself or other chemicals along with the synthesis? There's another study here is from group Antolin. They provided this carbon nanofibers. As you see here, you can see the TM images and SEM images, and there are, it's just really huge variation in size and length and so forth. And then it was my really interest to see how surface chemistry is gonna affect the behavior of these nanomaterials in the test tube. With the interaction with the cells. It is amazingly this change in surface chemistry. For example, glycine, and as you see from the graphs, modification with glycine and amine and glucose, and they all improve the toxicity of these materials, even though they are mass. But I mean, when we tested with the genotoxicity, it looks like that they are not, they are not very toxic. They are not really causing too much trouble for DNA damage. The other one is the graphene oxide nanomaterials, graphene oxides from upline nano solutions. A similar story here as well. Then also the Avanzare. Okay, this is obviously. I mean, we see, although we see a trend in the how toxicity improves with surface chemistry, there is a variation in the response. Finally, in ecotoxicity tests, I mean, there is again a lot of data here and a lot of different types of tests. And I'm not going to go through all of these the tests and results, but I'm going to show you just a few of them. For example, zebrafish embryo toxicity testing. And then, of course, here during the testing, the stability of the testing medium is really important. First, the Itané and INL, they studied the behavior of these nanomaterials, modified nanomaterials in different mediums, like fresh water and ultra pure water, and so forth. Then they tested these animals, and they found that there are some variations again in the results, and the glucose modified ones show more toxic behavior here for the Z test. The influence on the as you see here on the table, you can see a lot of different parameters are checked. And then this is something that is, interesting. And also the other test, for example, ALGA growth inhibition test, it obviously modified ones showing some effect on this case. This is interestingly. I mean, then because when it is compared to graphene oxide, but in Dafina acute immobilization test, they also, modified ones are making some impact on this toxicity, the picture landscape. But there is because I can tell you why this is so. Okay. Then also other tests, earthworm acute toxicity test and the bioaccumulation tests. So I mean, this is results are here is interesting because this the surface modification causing some effect. Sometimes, generating more toxic materials, sometimes generating less toxic materials. Actually, this is the reason why surface chemistry is an important point here to explore and understand, and along with the in vitro testing, because it provides a lot of data, okay, and can be used in the further studies. For example, in this project, work package 3 is concerned with the generating data from these testings and surface modifications and testing, and also literature, the data. Okay, in conclusion, I'm not going to, I think I'm almost done here, but it shows that surface chemistry is an important parameter to understand the how these materials behave. Okay, it is possible, very possible, to make nanomaterials less toxic nanomaterials using surface chemistry. And the diversity that is observed with the results is there are many different reasons for this. It could be nanomaterial itself, or it could be just the surface chemistry or the metrics these tests are performed. Okay, and in general, I mean, we see that the surface chemistry is a critical parameter to define the behavior of these nanomaterials in biological systems. And this in vitro testing is really, really important. And there is I would say, in some cases, a good correlation with the in vitro and in vivo results. Then I'm going to conclude my talk, and thank you very much for your time.